All right, in this video, I wanted to give a really brief and simplified overview of the process of DNA replication. So we'll think about a couple major steps in this pathway, how the structure of DNA is involved in this process, and so we'll go from there. Okay, so we're going to be thinking about DNA replication. And I want to point out a few key details before we move on. So first, this process of DNA replication is going to create two identical copies of our chromosomes, or DNA. All right, so that's the main point, is we're going to copy all the DNA inside the cell. But I want to point out a couple things that DNA replication doesn't do, or when we don't do DNA replication. So first, DNA replication only occurs during S phase of interphase. So it only ever happens once during the cell cycle. We talked about the cell cycle a while back. S phase is the only time in the life of a cell that the DNA is replicated. Okay. DNA replication also only occurs prior to cell division. So the only reason a cell ever replicates its DNA is because that cell is going to become two cells, and both of those cells are going to require a copy of the DNA. So using those two pieces of information, I'm going to say one thing DNA replication is not. It's not required prior to transcription or translation. So many students make this inappropriate association between DNA replication and these other processes, transcription and translation. Again, the only time we replicate DNA is when the cell is going to divide into two cells. And these other processes we'll talk about next, transcription and translation, do not require DNA replication prior to their occurrence. All right, so with that cleared up, let's think about the structure of DNA and how it relates to the process of DNA replication. So here's a diagram illustrating the structure of DNA. Hopefully this looks familiar. It illustrates the double helix structure of DNA. And I want to point out a couple important features. Mostly I want to point out how this molecule is held together. Okay. So I'm going to point out two kinds of bonds that hold this structure together in this particular shape. So the first kind of bond is a covalent bond. You'll remember from our discussion of bonds at the beginning of our course that covalent bonds are very strong. And the covalent bonds are found in the structure. They connect these chains of nucleotides together into these long strings. So each of these twisting strings is one chain of nucleotides held together by covalent bonds in that chain. Okay, so from top to bottom, this molecule is very strong. It's held together by these strong covalent bonds. Okay, the other kind of bond in this DNA molecule are hydrogen bonds. You'll remember from our discussion of bonds that hydrogen bonds are very weak. There are a lot of them in here, but each one of them is very weak. These hydrogen bonds hold the two chains of nucleotides together. So these linkages, side to side within this molecule, the linkages are called base pairs. They're held together by hydrogen bonds. And you'll remember that the base pairs are always A with T or G with C. So in our DNA molecule, we put together lots of strings of the letters A, T, G, and C in whatever order is required by the organism. Those strings are held together by the strong covalent bonds. Then we take two of those strings that are complementary to one another, and the complementary base pairs, A with T or G with C, hold those two strings to one another. And those connections are hydrogen bonds. All right. So top to bottom, we're very strong. Side to side, this molecule is relatively weak because it's only held together by hydrogen bonds side to side. So let's think about the process of DNA replication and why those structural elements are important towards that process. All right, So to discuss DNA replication, we're going to think about a couple important enzymes. In this video, we're just going to name two enzymes that are involved in the process. And the first enzyme is this enzyme named helicase. Helicase's job is to unzip the DNA, or to break apart the helix. All right. So I have a description here. We're going to copy the DNA, and in order to do that, helicase, this enzyme, breaks the hydrogen bonds, these weak bonds, holding the two chains of nucleotides together. So on this diagram, if I wanted to illustrate where helicase is and what it's doing, I would highlight this area right here. And I would tell you that helicase just broke apart this GC complementary base pair, unzipping the DNA. Next, helicase will break apart this AT complementary base pair. And then helicase will keep moving upwards on this DNA molecule, unzipping these complementary nucleotides as it goes. So thanks to helicase breaking these hydrogen bonds, we separate these two strands of DNA, and then using the nucleotides that are exposed, we can build a new complementary string next to it. So that's our next step. So helicase opens the DNA up, and then our next enzyme, DNA polymerase, builds a new strand of DNA that matches one of these strands that we have exposed. So the enzyme DNA polymerase reads the original DNA strand and builds a new complementary strand. So if I wanted to show again on the image here where DNA polymerase is acting, I would highlight this spot right here. And in this case, DNA polymerase is looking at this unzipped piece of DNA where there are these unpaired nucleotides, C, T, and then another C. And it's going to grab and then place the appropriate complementary nucleotide into a new strand right across from it. So DNA polymerase is going to put a G nucleotide into this position across from the C and attach it to this new strand of DNA that's being built. So DNA polymerase just put these two T's into the new strand. Next it's going to put a G, then it will put an A, then it will put another G. And DNA polymerase will keep working its way up this strand, following helicase, copying the DNA, making a new complementary strand. This is happening on both sides of the molecule. So if we look at the left-hand side of this unzipped DNA molecule, we would find DNA polymerase right there, adding some new nucleotides to the new strand. And there would be another DNA polymerase molecule right there, adding new nucleotides to that growing DNA strand. All right? So through the action of those two enzymes, helicase and DNA polymerase, we can separate our two strands of DNA and then build new copies. And the products would look like this. So here's our original strand of DNA. Again, helicase would pull these two strands apart by breaking the hydrogen bonds. And then DNA polymerase would build these new golden strands of DNA that are complementary to our original parental strand. And at the end of the process, at the end of DNA replication, we will have two identical DNA products. They're identical to one another, and they're identical to the beginning product. So the products of DNA replication, they're identical to one another. They have the exact same information. And then the other thing we want to point out is that each of these products of DNA replication contain one old strand, the blue color piece of DNA, and one new strand, the more golden piece of DNA. And so each molecule is half old and half new. And that characteristic, being half old and half new, is why we call this process of DNA replication semi-conservative, because we save half the old information and make a new complementary strand to match it. So half old, half new. That's why we call DNA replication semi-conservative. All right, the last piece, couple pieces of information that I want to point out, is that we've drastically simplified DNA replication. I told you about DNA polymerase. So here's a DNA polymerase enzyme right here, and another one right here. And I told you about ligase right here that's breaking open the helix. But DNA replication actually requires a number of other enzymes and proteins and some small RNA molecules in order to work correctly. And so in certain biology classes, you might need to know that detail. In an intro non-majors biology class, we're not going to go into that much detail. Okay? But I do want you to understand that there are many molecules involved in this process, and it is a complex process. But for our purposes, understanding that helicase opens the DNA, and then DNA polymerase reads the old strand of DNA and creates a new strand of DNA to match it, then we understand what we need to about DNA replication. All right, the very last point I want to make about DNA replication is that the process begins at a certain spot in our DNA called the origin. So here I'm showing two strands of DNA. The complementary strands are different colors. And then right here, we've started DNA replication. So DNA replication began at this spot right here in the middle, and that's the origin. As we continue DNA replication, these new dotted strands are going to grow to the left and to the right. We're going to see this. So what we call a replication bubble is growing side to side. But because we have so much DNA to replicate, we don't just start in one spot. Somewhere else in our DNA, actually many other places in our DNA, there will be additional origins of replication. So here I'm showing a second origin, where we begin DNA replication right there as well. And this replication bubble will grow left and right while we continue the process. So we're going to keep copying the DNA, making the dashed or dotted strands of DNA, the new strands, until they run into one another or reach the end of the DNA molecule. And when they do, we'll have two identical copies of the DNA molecule, each one consisting of one old strand, the solid line, and one new strand, the dotted line. So in organisms that have a lot of DNA, we begin copying at many origins so that we can copy the DNA more quickly, rather than trying to start in one spot and copy it all from there. So that's the last detail I want to say right now about DNA replication. As always, let me know if you have questions, and I'll talk to you soon about some of the other processes involved in the central dogma of molecular biology.